There we go.
Okay, so I'm going to share this screen and we're sharing, right?
Mm-hmm.
Okay, and let me put on, I'm hoping I'm going to share the right, see the projection and not the presenter, right?
It looks great.
Okay, good enough. So I much prefer this to be interactive and people to ask questions. I do not do very well talking and reading at the same time. So I'm going to ask Sandra and Todd to do me a favor. And if questions come up during the presentation, for you to stop me and read them out loud. But I encourage all the attendees to feel free and just talk. And I promise I will stop talking. But I just keep going. So I prefer when people stop me in the middle. Better to talk about things when you have questions. And the other thing is, you probably say this to your own students. The only bad questions are the ones that are not asked. So don't let them stay in your head. Let them come out your mouth and share them with us. So I was asked to talk about antibody validation, which is a topic that most of us don't like to talk about, but is exceedingly important and is a never-ending process. So what is antibody validation? And it's the process by which we prove what an antibody binds to. It is always an imperfect process, and depending upon how well you are able to validate your antibody, you have a certain degree of certainty, and you want to pay attention to what your caveats are, because nothing is going to be perfect in the process. And in some respects, it boils down to validation is a really good set of controls. So why is this important? Probably the early 2010s, science had a very public crisis of reproducibility. There was a big book written on this, and I think it was in the New York Times, and it was really bad. And one of the factors that played into this was the irreproducibility of experiments that involved antibodies. And as Sandy told you earlier, antibodies are important reagents in a broad spectrum of techniques used in biomolecular research. And so when we're using antibodies that are not properly validated, that's one aspect of how antibodies were part of the problem. We come to the wrong conclusions. We get irreproducible results, and we waste a lot of tax dollars and a lot of philanthropy. So much so that even now we are still having literally webinars on how to validate antibodies. And personally, as a scientist, I find this exceedingly embarrassing. People have to have talks on how to run controls. And I think part of this is that people think of antibodies like nails and screws as you walk down the aisle of Home Depot. There's just tons of them. There are billions of antibodies out there. And it's a little reagent. It's inconsequential. And the problem is, unfortunately, it's not. You're not going to use a concrete nail to put the shingles on your roof. You have to use the right antibody under the right conditions and know what you're using. So what do antibodies bind? And I think you got a really good introduction to this. So I don't really have to go into too much, except that I want to point out that the paratope of the antibody binds a three-dimensional surface. That epitope is a three-dimensional surface, okay? Linear, discontinuous, whatever it is, but it's three-dimensional. It happens to be on the target, But if that three-dimensional surface is on another protein, let's say a family member, and they're family members because they're conserved, and that is the conserved region that makes those 2 or 5 or 10 proteins all similar, that antibody will bind every single family member. And family members tend to be expressed in the same cells or in a family of cells or different cells or different times. So that antibody will bind different things at different times. If the epitope, that three-dimensional surface, is slightly different on a different protein, the antibody will bind it as well. It will just bind it at a lower affinity. So you have to be really careful about understanding what your antibody is binding. Antibodies in general, and because I make monoclonal antibodies, I know this, looking at the single protein, antibodies can be very sticky in general. I have no clue why, to be quite honest. I should ask one of the structural biologists that I work with and that use the antibodies in cryo-EM. Why? But you can get very sticky antibodies. And everything that I tell you today is going to be true for monoclonals or polyclonals. The polyclonals are much more confusing because you will have antibodies against multiple three-dimensional surfaces. So scientists are really sloppy in how they speak. And that's because sometimes the names get really long and, everyone wants to say things quickly. So this is a particular name of an antibody, and we would call this an anti-insulin monoclonal antibody. But if we wanted to be precise, we would actually give a name to the epitope on insulin that it binds, because the antibody is actually against that epitope. It is not against insulin. Insulin just happens to have that epitope. And if we wanted to be more precise, we would say that it's also against biosimilars. So if there were slight changes in that surface, that antibody would also bind it with a lower affinity. And I'm sorry that I'm pounding this home, but it's very important in validation that you understand that the antibody is not against a protein or a small molecule or an amino acid that's phosphorylated. It is against a three-dimensional structure, because if you understand that, you're going to think about the right things and you're going to come up with the best controls possible. Not all, not perfect, but the best ones you can do. Okay. Does anyone have any questions so far? Okay,
Yeah,
we're all really, go ahead.
Yeah, so how do we find these similar surfaces, right? So you can't use simple sequence analysis, can you?
No, you can't. To the best of my knowledge, the only way you can really do it is through a structural. So at this point in time, I'm not quite sure people are doing X-ray crystallography that well or that much. Cryo-EM is probably the way you can do it. There's supposed to be a way where you actually have the complex and then modify the protein around it. Someone really wants to know, I'll go and find out. But that doesn't give you the three-dimensional surface. That just tells you the amino acids that are involved. But to get the structure, you actually have to do cryo-EM or X-ray crystallography. And normally there's about 3 to 5 amino acids that are key for the binding. It will cover about 12 to 15, but 3 to 5 are usually the key amino acids. If you change the others, you'll probably just change the affinity a little bit.
Francis,
Yes.
what kinds of interactions are most important with those 3 to 5? Are they usually ionic? Are they hydrophobic? Or
Oh, yes. So the paratope is floating around in an aqueous solution. So that is charged. So you want the epitope and the paratope need to be hydrophilic, right? Hydrophobic areas are going to be within your target and within the antibody. So charged areas are what, if you're going to choose something to make an antibody against, you want to choose charged areas. Hydrophobic regions of the protein do not make good immunogens. Oddly enough, prolines are pretty good. Alanines and glycines are useless because they have very little structure to them. Phosphorylation sites are great because that phosphate has got a huge amount of charge to it. I can always help someone make choose areas, but to be quite honest, the 2 speakers ago, and I apologize that I don't remember your name if you're still here, that database is pretty good, actually very good. You can use that if you're going to use portions of it, but if you're going to choose, if you want a continuous epitope, which are usually important for flow cytometry, immunoprecipitation, stuff like that, you're going to want to choose a whole domain, right? And not break it up. I make monoclonal antibodies for a living, so I'm always choosing areas with which to make, with which to immunize the animals.
Thanks. And that's IEDB is the database.
Yes,
and Marcus
the IEDB.
told us about it.
Thank you. Okay, so what is antibody validation? Going back to an earlier slide, it's the process by which one proves what the antibody binds, irrespective of what you think it should bind. So you need to prove that it binds the target, right? So if you're developing a brand new antibody, does it actually bind what you want? Or which of all the antibodies that you made binds your target? And prove that there is no off-target binding, unless you want it. You want one that crosses all of the family members. Maybe that's what you want. So if it's off target, does it bind to an identical epitope on another protein or to a similar epitope? You want to make sure, you absolutely don't want this. You want no non-specific binding. This is something I haven't done, but this is very important. If I want an antibody that works in Western blot, I will be testing it in Western blot with a specific protocol and a specific sample. If I change my protocol or I change my sample, I have to revalidate everything because it is protocol and sample specific. Or if I'm doing immunohistochemistry, if I change from spleen to muscle, or I change my fixation protocol, all the epitopes are up for grabs. And I have to revalidate because those 3-dimensional structures change. The proteins in those tissues change. And it really, I'd like to use a 3-4-letter word, but I won't, I'll be good. But your previous validation processes will help inform what to do now. The epitopes that are in for flow cytometry are different than ones for Western blot. The epitopes available for flow cytometry will be similar to the ones that are available for immunoprecipitations, but they may not necessarily be the same. So remember that the validation is dependent upon the application, the protocol, and the sample. These two, because the epitopes that are available are going to be dependent upon how the antigen has been prepared, and the sample usually because what proteins, we'll talk about proteins because they're the most common target, what proteins are available, difference between spleen and muscle, okay, for example, or species maybe. So after this whole embarrassment about science and realizing that antibodies were one of the key things that we could fix and were also the cause behind all of this. Oh, there were meetings and congresses and working groups and everything. It was really a whole hubbub about this. And these are just 3 different things that came out of it. And there were a few more papers and everything. I'm going to go over, I'm going to show you kind of what I like from the 3 of these. Let's put it that way. And kind of using this as a backbone. And Sandra and Todd, I will send you a link for this. So this comes from a very prominent journal by the name of Cell, who is working with a company called Cell Signaling Technology, a very respected developer and distributor of antibodies. And it talks about this in rather plain English, but much more detail than I'm going to provide you right now. Okay, so they provide different strategies, which is what they're trying to symbolize here. So the first strategy that they provide is a binary strategy. So kind of zeros and ones, on and off. And they're looking at endogenous expression, which is really nice because it's natural, right? We're looking at the normal levels of the protein in the animal, in the human. Not that we're not animals, I guess we're animals too. In the cell line, if a cell line is considered natural, but scientists tend to say it's natural. Or in knockout animals, in animals or cell lines that have been modified by CRISPR and the gene has been taken away. In others where it's been knocked down, or you can maybe turn it off or modulate it under certain conditions by growth factors or confluence, or confluence. So basically you want an on-off situation, so it's black and white, right? But in these guys, your control is that you need to confirm it independently that the protein is not there, okay? Usually you would do that by monitoring for the presence of RNA, because if there's no RNA, there's no protein. You could also do it with another antibody. So here's an example. This is basically immunohistochemistry, but done with cell pellets. So instead of tissue, they took a cell pellet and they embedded it in parafilm, did the sectioning and did the staining. So they took Jurkat cells and they took RL cells and stained for Helios, which I actually don't know what that is, but you can see it here and you can't see it here. It would have been a much better assay had they actually knocked out or knocked down the Helios in the Jurkat cells, as opposed to looking at a different cell line, because then we would have known that the staining was not to any other protein in the Jurkat cells, but not a bad assay. This one is better, like I said, sorry, let me take off my glasses. The RKIP cells here, the MEF cells, the mouse embryonic fibroblast cells in their wild type form, or where RKIP has been knocked out, you can see that the protein is present in the wild type. Sorry, it's I'm touching my pad, but it's not present when it's knocked out. So this is a much cleaner thing, and that antibody has now been validated for these cells, for MEF cells in Western blot. Okay. And one would hope that that would be true also for other cell lines, right? So now you're like 10 steps ahead for another cell line in Western blot. It's very likely that that will work. And you see a little binding, but you see you've got binding here in the HeLa cells and the COS1 cells. And this is non-specific binding. Or these proteins also have the linear epitope for this. And usually scientists just accept that and move forward. That's the caveat. So here's another one. So instead of on, off, black, white, we do a range, right? So maybe we have samples that have different levels of expression, and it correlates with the levels of RNA that are in it, right? And you have to keep in mind that the level of RNA doesn't necessarily mean that they have different levels of protein. Scientists accept that caveat because if you have different levels of RNA, you also have the regulation of the protein. It could be getting degraded, but we accept that caveat. We keep that sitting in the back of our head, okay? So these samples have a range of endogenous protein, and you can regulate them naturally. So different levels of a hormone. So in this particular case, and unfortunately, this is I guess it is different levels. So you have an endogenous level of phosphorylated C-Jun, okay, at serine 73. So there's some baseline level, okay? And I would presume that if they had a mutant that had no serine 73 that was changed to an alanine, that you would have no green. They don't have that control here. And then when they treat it with anasomycin, they get a great level of phosphorylation. And so that proves that this antibody detects the phosphorylation state of C-Jun at serine 73. And I have to take off my glasses. I apologize. And this one, yeah, this one isn't good. This one is worse because this one doesn't show ranges. I guess it does to some degree because you would normalize for the amount of actin. But in the interest of time, I'm not going to worry about that. So this one is called orthogonal. And here you're comparing the data in your experiment, your antibody experiment, with other data from that same sample, right? So if I'm looking at Jurkat cells, I need to be looking at the protein analysis in those same cells, but from a different perspective that is independent of antibodies. The one I really like is in situ RNA, where you're literally binding RNA, let's say in tissue, like that immunofluorescence, and you're binding the RNA, and you can see in the same cell that that antibody and that RNA are matching. So in this particular case, they're looking at RNA levels across a lot of cell lines, and these two are RT4 and HLDM1. And this one is the amount of RNA in RT4, and this is the amount of RNA in HDLM2, and you would expect a lot of staining here and no staining here. So this is an example of orthogonal, which is, I guess, transcriptomics. The other one is a multi-antibody strategy where you're using 2 antibodies to confirm. So you take a well-validated antibody or set of antibodies, and they're used as benchmarks to prove that the unknown antibody is indeed binding the target. You have to make sure, if you're doing it in the same technique, that the antibodies don't bind the same epitope, basically that they can bind simultaneously. It's a great approach, but you, when you're using a recombinant strategy that I'll show you in the next slide, but you should never use it as the only strategy. It's a good combinatorial one. And so basically in this particular case, they immunoprecipitated with one of them. I forget whether it's the known or the new one. And then what they did, ran it on a gel and did a Western with the other one. And the only way that this would come up positive is if they both bound new 2 or new N, I forget. So the recombinant strategy is one that is used a lot because it's easy, right? So you take a cell line and you overexpress your target. So your cell line without it is your negative control. Your cell line with the recombinant protein is your sample where you want to see staining. It's very clear, it's black and white. The problem is it's not natural, but it has some strengths because you can change how it's expressed. You can do mutations to it. In this particular case, they put a MIC tag on it, to which we have lots of MIC antibodies. And we can see that that our test antibody co-stains with it. And you can see that they overlap perfectly. In the interest of time, I'm not going to go over the Western blot. Sorry, this company does a lot of Western blot and flows and IHC and IF. So there's other ways to do this. So you could, if you're doing immunoprecipitation, you could do a protein identification of the protein that the antibody brings down by mass spec. And then there's no doubt that that antibody is binding that target. Or if you have multiple antibodies, multiple proteins that come down, you can see what proteins the antibody is bringing down. You could do competition. You could do co-localization. So if we had this antibody that I know bound in, let's say, the mitochondria, and we knew our protein should be in the mitochondria, and this was mitochondria, it's not obviously, but so co-localization in a tissue or a cell correlates. That's another way of validating one. Protein arrays can tell you epitopes, other proteins that might bind. Anything to substantiate that the protein does or does not bind your target or another target. So in the end, validation is really just all about controls, and more controls, which doesn't sound fancy, sounds really boring, but unfortunately, like exercise, is very important. And the two times, and the two times when it has to be done is when the antibody is initially developed, and then hopefully, and this is when we run into reproducibility problems, bad conclusions in our experiments, is when each experiment is run. And that's because the epitopes that are available, the three-dimensional surfaces to which the antibody binds, is technique dependent, protocol dependent, and sample dependent. And I really hope you don't feel like this. I am always available to answer questions. I don't expect you to necessarily write this email address down. You can always get it from Todd and Sandra. But do you have any questions?
Oh, will anybody want to touch antibody?
I really don't want to scare you away, but
So,
I like your passion. I really do. And I like the fact that you're very thorough in stressing what's important for us to remember.
thank you.
Francis, I go to Antibodies R Us and I buy some antibodies.
Yes.
Do they tell me the ways in which I should use that antibody? So how they validated the antibody? Because I think that was a source of that reproducibility issue.
Yeah, so they should tell, well, minimally they have data, and they should tell you how they do it, but a lot of times they're just distributors and they don't actually know how it was validated. And they were just given a figure and some minimal information, but they won't have, what they won't have is they won't have the protocol. They might have known that they were HeLa cells or Jurkat cells, and they definitely won't have the technique. Well, I guess they'll know it was a Western blot. And a lot of things are just validated by Western blot or flow cytometry if it's a flow, but they almost will never have the protocol. And the other thing is, since many of them are distributors, they're all selling the same antibody.
But you don't know that.
which you don't know unless they're all putting up the same figures. So you need to
Got it.
compare the figures.
Wow.
Yeah.
There is a database about antibody validation. I can't remember the name of it at the moment, but I know there is one that exists.
There's actually an online open access journal for antibody validation. So if you do it for a technique, for a protocol, for a sample type, you can actually publish just that data. And you can always refer to it, because most people, bury it in the supplementals, because that's where I would put it, right? You only have so much room in your manuscript. And it's kind of lost forever, so to speak, because you're going to refer to it, but no one's ever going to look at it. Where if you put it in a front, in front and center in one manuscript, people might actually go look at it.
No, that makes a lot of sense. Thanks. Are there other questions? Okay, well, thank you, Francis. We really appreciate it.
Have fun with your hackathon.
Thank you. I'm going to stop recording.